Welcome to the GNP Sir Classes official YouTube channel. In our today's online class, Professor Govind Narayan Purohit from GNP Sir Classes explains general pathology. Watch this video completely to learn in detail. Here are the contents of today's class that includes topics like causes of disease, hemodynamic disorders, cell death, gangrene, calcification, and immunopathology. First, let us define pathology, which is the scientific study of diseases in animals, focusing on the structural and functional changes that occur within their bodies. In its shortest form, pathology is the scientific study of disease. Pathology is essential for accurate diagnosis of diseases. It also helps in understanding how diseases develop and progress that may eventually help in developing effective treatments and preventive measures. Pathology has many branches such as anatomical pathology, histopathology, cytopathology, surgical pathology, clinical pathology, hematology, and clinical chemistry. First, Professor Purohit mentions the scientists in vet pathology. Hippocrates, 460,370 BC, is considered the founder of scientific medicine, father of Western medicine. He gave the humoral theory of diseases. He had a great interest in anatomy and pathology. Aristotle, 350 BC, started animal dissections and described anatomical structures. Claudius Galen, 130 AD, followed the humoral theory of diseases and proved that arteries contained blood. Avenzoar, 1091-1161, was the first physician to make post-mortem dissections. Antonio Benavini, 1443-1502, used anatomic dissections to determine the cause of death and introduced necropsy to the medical field. Andreas Vesalius, 1514-1564, was the founder of modern human anatomy. Percival Pott, 1714-1788, was the first scientist to demonstrate environmental carcinogens. Giovanni Battista Morgagni, 1682-1771, father of modern anatomical pathology. He published Seats and Causes of Disease in 5 volumes. Edward Jenner, 1749-1823, discovered vaccine and is called the father of immunology. Louis Pasteur, 1822-1895, germ theory of disease. Robert Koch, 1843-1910, causal relationship between microbe and disease. Karl von Rokitansky, 1804-1878, autopsy. Rudolf Virchow, 1821-1902, father of microscopic pathology, cellular pathology, modern pathology. First to describe thrombosis, embolism, leukemia. He coined terms like chromatin, neuroglia, agenesis, parenchyma. Shalihotra, 2500-1500 BC, first known veterinarian of the world. Charak, 800 BC, wrote Charak Samhita, with details of the cause of diseases and impact of the environment. Renatus Vegetius, 1749-1823, father of veterinary medicine. Jean Fernald, 1497-1558 AD, first to attempt to codify the knowledge of pathology. Xavier Bichat, 1771-1802, ad., father of histology and pathological anatomy. John Hunter, 1728-1793, ad., first experimental pathologist. J. Kornheim, 1821-1902, originator of modern experimental pathology. Foundation for the pathology of inflammation. Introduced frozen sections. E. Mechnikov, 1884 AD, phagocytosis. Papanicolaou, 1883-1962 AD, father of exfoliative cytology. Now let's talk about the causes of disease. Disease can be caused by intrinsic and extrinsic factors. The intrinsic factors include genetic defects, inherited disorders like hip dysplasia in dogs or immune disorders, allergies, and autoimmune diseases. The extrinsic causes of disease include infectious agents like bacteria, viruses, and parasites, poisoning from plants, chemicals, or medications, trauma from accidents or fights, and environmental factors such as poor air quality, inadequate nutrition. Now let us talk about the hemodynamic disorders. Hemodynamic disorders are disorders pertaining to the altered dynamics of blood flow. This include hyperemia, which is increased blood flow to an organ, congestion, which is passive accumulation of blood in tissues, and hemorrhage, which is escape of blood from the blood vessels. This also includes edema, which is abnormal accumulation of fluid in the interstitial spaces. Other hemodynamic disorders include thrombosis, which is formation of a blood clot within a blood vessel, embolism, which is obstruction of a blood vessel by a foreign object, and infarction, which is an area of tissue death due to lower blood supply. Let us discuss the terms further. Hypostatic congestion is the accumulation of blood in organs and tissues of the lower side of a recumbent animal due to gravitational flow. This can occur due to cardiac diseases that lead to sluggish movement of blood. White circle, prolonged recumbency. White circle just before death. White circle most common in lungs, thin-walled capillaries. Another term is ischemia, which means local deficiency of arterial blood in an organ and can occur due to blockage from within. White circle arterial wall defects. Atheroma, abnormally increased muscle tone as seen in ergot poisoning. White circle pressure from outside. Now let's discuss hemorrhage. Hemorrhage is the escape of blood from the lumen of arteries, veins or capillaries is called a hemorrhage. It can be white circle external hemorrhage if the extravasated blood is visible outside the body, or white circle internal hemorrhage if the extravasated blood is visibly found or accumulated in body cavities or tissue spaces. Based on the extent, hemorrhages can be classified into white circle petechiae are small pinpoint hemorrhages in serous surfaces, mucous membranes and body surfaces common in septicemic diseases. White circle ecchymosis, extensive hemorrhages in tissues or body surfaces, usually larger than 1 cm in diameter. White circle extravasation are large extensive hemorrhages in tissues. In relation to hemorrhage, let us understand some terms. Epistaxis means hemorrhage through the nostrils, hematemesis means regurgitation of stomach contents mixed with blood, or blood only from the mouth. Hemoptysis is a medical term that refers to coughing up blood from the lower respiratory tract. Metrophagia and enterorrhagia means abnormal bleeding from the uterus and intestine respectively. Whereas hematuria is the blood in urine. The terms hemothorax, hemopericardium, hemoperitoneum are used for accumulation of blood in the thorax, pericardium, and peritoneum respectively. A hematoma is a localized collection of usually clotted blood that forms in a tissue, organ, or body space as a result of a broken blood vessel. Purpura, also called blood spots or skin hemorrhages, refers to purple-colored spots that are most recognizable on the skin. The term apoplexy means unconsciousness or incapacity resulting from a hemorrhage in the brain. Now let's talk about edema. Edema is an abnormal accumulation of fluid, either transudate or exudate, in the intercellular spaces and body cavities. When this fluid accumulation is in the fallopian tube, pericardium, or meninges of the brain, the terms hydrosalpinx, hydropericardium, and hydrocephalus are used. The macroscopic features of edema are I. It is swelled, cold and smooth to touch. The swelling pits on pressure. The microscopic features are also shown above and include the pink and granular nature of fluid with widening of intercellular spaces. There can be different types of edema including inflammatory edema, cardiac edema, renal edema, pulmonary edema, myxedema, angioneurotic edema, and Brisket disease. Next we discuss thrombosis, which thrombi is an intravascular clotting of blood. There are different types of thrombi, each with a specific cause and effect, which are shown here. The pathogenesis of thrombus formation is also shown and the fate of the thrombi are also shown for understanding. Next is embolism. Embolism is the mechanism by which solid, liquid and gaseous foreign materials are transported through the circulatory system. The foreign material circulated is embolus, emboli. Emboli are usually lodged in arteries and capillaries as their lumen get progressively narrowed from the heart to the supplying organ. There can also be venous emboli, usually do not get trapped in organs other than lungs. Lymphatic emboli, trapped in the sinuses of lymph nodes. Lymphatic emboli, filarid worms, occlude lymphatics, and result in a disease elephantiasis. Fat emboli, are usually lodged in the lungs. Air emboli, cause casein disease, decompression sickness, bends. Parasitic emboli, Dirofilaria immitis worms act as emboli in the pulmonary artery of dogs. Schistosoma nasal, occlude the nasal blood vessels. Schistosoma mansoni, occlude the mesenteric blood vessels. Other types of emboli include tumor emboli, bacterial emboli, and thrombotic emboli. Now let's consider what are the cellular changes that occur in response to a disease. They include 1. Glycogen overload, abnormal accumulation of glycogen within cells. 2. Amyloidosis, abnormal deposition of amyloid protein in tissues. And 3. Fatty changes, accumulation of fat within cells. When cells are attacked by pathogens, they may cause reversible or irreversible injuries to the cells. This figure explains some of the biochemical mechanisms of cell injury. Cell death is the termination of a cell's life. Two primary types of cell death are evident: necrosis, unregulated cell death due to injury or disease. Apoptosis, programmed cell death, a controlled process essential for development and tissue homeostasis, e.g., removal of unwanted cells during embryonic development. Necrosis refers to a sequence of morphological changes that follow cell death in living tissue or organ. Nuclear changes, karyolysis, the basophilia of the chromatin may fade. Pycnosis, characterized by nuclear shrinkage and increased basophilia. Karyohexus, pycnotic nucleus undergoes fragmentation. Cytoplasmic changes, the necrotic cell is pale, ghost-like appearance. Necrotic cells become individualized and may slough from the epithelium. Necrosis is of the following types: coagulative, protein denaturation, e.g., myocardial infarction, heart attack. Liquefactive, enzymatic digestion, e.g., brain abscess. Caseous, cheese-like appearance, e.g., tuberculosis. Gangrenous, necrosis with superimposed infection. Apoptosis. Apoptosis is a pathway of cell death that is induced by a tightly regulated suicide program in which cells activate enzymes that degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. Apoptosis results from the activation of enzymes called caspases. Cysteine-dependent aspartate-specific protease, caspases. Initiator caspases are caspases 2, caspases 8, caspases 9, and caspases 10. Executioner caspases are caspases 3, caspases 6, and caspases 7. Apoptosis may be divided into two phases: initiation phases, activation of initiator caspases. Two distinct but convergent pathways: the intrinsic, mitochondrial, pathway; the extrinsic, death receptor initiated, pathway. Execution phase. Activated initiator caspases activate executioner caspases to cause cell death. Mechanism of apoptosis. Normally anti-apoptotic proteins prevent apoptosis, prevent leakage of cytochrome C into the cytosol by keeping the mitochondrial outer membrane impermeable. Right-pointing arrow prevent apoptosis. These include BCL2, BCLXL, and MCL1. The pro-apoptotic proteins form a channel on the outer mitochondrial membrane, and when there is leakage of cytochrome they activate caspases and induce apoptosis. Some other proteins called sensors sense the cellular STRSS and damage and regulate the balance between the anti- and pro-apoptotic proteins. These include BAD, BEAM, BID, PUMA, and NOXA. Now let's understand the changes after death called post-mortem, PM. Alga mortis is the cooling of the body. It commences at or before the stoppage of blood flow. Rigor mortis is the contraction of muscles after death. Important for estimating the time of death. Livor mortis, hyperstatic congestion is, due to gravity, accumulation of blood in vessels of organs that are found on the lower side of the recumbent animal. PM clot is the coagulation of blood in the vessels after death. Chicken fat is the white clot while the current jelly clot is the red clot. Imbibition of hemoglobin. PM staining is pinkish discoloration of the endothelium of larger vessels due to hemoglobin, liberated from lysed erythrocytes, after death. PM there is displacement of organs. PM there is rupture of organ and tissue. Now we consider the term gangrene. Gangrene is a serious condition where a part of the body's tissue dies. It's typically caused by a severe lack of blood supply to the affected area, often due to injury, infection, or underlying health conditions like diabetes. It is of the following types: dry gangrene, tissue becomes dry, shriveled, and dark, e.g., extremities with poor blood supply. Wet gangrene, tissue becomes edematous, discolored, and foul-smelling, e.g., internal organs with bacterial infection. Gas gangrene, gas-producing bacteria infect necrotic tissue, e.g., clostridial infections. Let's try to understand pigments and their significance in veterinary pathology. Pigments are colored substances that can be either endogenous, produced within the body, or exogenous, originating from outside the body. Pigments provide valuable clues about underlying diseases and are useful in disease diagnosis. They can help in understanding the disease processes and can be useful in evaluating the severity of a disease or its severity. Pigments can be exogenous or endogenous. Exogenous pigments are pigments originating from outside the body. Examples: carbon, black pigment from inhalation, e.g., anthracosis in lungs. Dust, inhalation or ingestion of various dust particles, e.g., silicosis. Tattoo pigments, deliberately introduced. Endogenous pigments are pigments produced within the body. Examples: melanin, brown-black pigment produced by melanocytes. Lipofuscin, wear and tear pigment, accumulates in aging cells. Hemosiderin, iron-containing pigment derived from hemoglobin breakdown. Bilirubin, yellow-brown pigment from heme breakdown. Calcification is the abnormal deposition of calcium salts in tissues other than bone. It's a common pathological process that can occur in various forms. Metastatic calcification is the deposition of calcium salts in normal tissues due to hypercalcemia. Causes: hyperparathyroidism, renal failure, vitamin D intoxication. Gross appearance: white, gritty deposits. Dystrophic calcification is the deposition of calcium salts in necrotic or degenerating tissues. Causes: necrosis, inflammation, and aging. Gross appearance: white, gritty deposits. Photosensitization is the skin damage caused by sunlight due to the presence of photosensitizing substances in the blood or tissues. In other words, photosensitization is the activation of photodynamic chemicals on the skin. Necrosis and edema are produced due to the release of reactive oxygen species, leading to mast cell degranulation and production of chemical mediators of inflammation. Photosensitization is the skin damage caused by sunlight due to the presence of photosensitizing substances in the blood or tissues. There are three types of photosensitization. Type 1 or primary. This occurs due to plant-derived photosensitizers ingested by animals. Type 2 or congenital occur due to genetic defects in porphyrin metabolism. Type 3 are hepatogenous, which occurs due to failure of liver to metabolize normal or abnormal pigments. Different names are given to the disturbances in growth of cells, tissues or organs, which are explained here. Aplasia, complete absence of an organ or tissue. Hyperplasia, incomplete development or underdevelopment of an organ or tissue. Atrophy, decrease in size of an organ or tissue due to loss of cells. Atrophy is physiologic with normal aging, whereas it is pathologic when it occurs due to disuse, denervation, ischemia, malnutrition. Hypertrophy, increase in size of cells, resulting in an increase in organ size. Hyperplasia, increase in the number of cells in an organ or tissue. Metaplasia, reversible change in which one differentiated cell type is replaced by another. Dysplasia, abnormal growth and differentiation of cells, often a precursor to neoplasia. Now we discuss an important topic in pathology, that is inflammation. Inflammation is a complex biological response of vascular tissues to harmful stimuli, such as pathogens, damaged cells, or irritants. It is a protective mechanism that aims to eliminate the cause of injury, clear out necrotic cells and tissues, and initiate tissue repair. The cardinal features of inflammation are rubor means redness, tumor means swelling, cal means heat, and dollar means pain. Aulus Celsus, 30 BC and 45 AD, described the main four signs of inflammation. Whereas Rudolf L. C. Virchow, Claudius Galen, described the fifth cardinal sign of inflammation, disturbance of function. Inflammation can be acute or chronic. In acute, there is a rapid onset and characterized by neutrophil infiltration. The patterns of acute inflammation are serous, catarrhal, fibrinous, suppurative, and ulcerative. In chronic inflammation, there is prolonged duration, characterized by lymphocyte, macrophage, and plasma cell infiltration. In response to attack by some pathogen, different cells migrate to the site. Chemotaxis aids in the recruitment of immune cells to sites of infection or inflammation. For instance, chemotactic gradients guide neutrophils to bacterial infections while driving T cells towards infected tissues. The chemotactic cells include neutrophils, lymphocytes, plasma cells, eosinophils, and mast cells. There are many other chemotactic factors that are shown here. The mediators of inflammation include cytokines, chemokines, prostaglandins, leukotrienes, histamine. An inflammatory cytokine or pro-inflammatory cytokine is a signaling molecule, a cytokine, secreted from immune cells like helper T cells, Th, and macrophages, and certain other cell types that promote inflammation. They include interleukin-1, Illinois-1, IL-6, IL-12, and IL-18, tumor necrosis factor alpha, TNF-alpha, interferon-gamma, IFN-gamma, and granulocyte macrophage colony-stimulating factor, GM-CSF. Inflammation may also have systemic effects like fever, leukocytosis, and acute phase protein production. The inflammation is terminated by switch from pro-inflammatory to anti-inflammatory metabolites. Anti-inflammatory cytokines include Illinois-10, TGF. Anti-inflammatory mediators are resolvin and protectin. Now we discuss wound healing. Wound healing is a complex biological process that repairs damaged tissue after an injury. It is a natural response of the body to restore the integrity of the skin and underlying structures. Key phases of wound healing are hemostasis. This is the initial phase where bleeding is stopped. Blood vessels constrict, platelets aggregate to form a clot, and fibrin is formed to stabilize the clot. Inflammation. This phase involves the recruitment of white blood cells, neutrophils, macrophages, to the wound site to clear debris and fight infection. Proliferation. This phase involves the growth of new blood vessels, angiogenesis, the formation of granulation tissue, a specialized tissue rich in collagen and blood vessels, and the migration and proliferation of epithelial cells to cover the wound. Remodeling. This is the final phase where the collagen in the granulation tissue is remodeled, leading to the formation of a scar. Wound healing occurs either by primary or secondary intention. Primary intention occurs in clean, incised wounds with minimal tissue loss. The wound edges are brought together, and healing occurs rapidly with minimal scarring. Secondary intention occurs in wounds with significant tissue loss. Healing involves the formation of granulation tissue and contraction of the wound edges, resulting in a larger scar. Here are some of the factors that affect wound healing. Severity of the injury. Larger wounds take longer to heal. Infection. Infection can delay healing and increase the risk of complications. Nutrition. Adequate nutrition, especially protein and vitamin C, is essential for wound healing. Blood supply. Good blood flow to the wound is crucial for delivering nutrients and oxygen and removing waste products. Age. Wound healing slows down with age. Chronic diseases. Conditions such as diabetes and chronic kidney disease can impair wound healing. Growth factors are relatively small and stable, secreted or membrane-bound polypeptide ligands, which play an important role in proliferation, differentiation, angiogenesis, survival, inflammation, and tissue repair, or fibrosis. Epidermal growth factor, EGF, stimulates epithelial cell proliferation. Platelet-derived growth factor, PDGF, stimulates fibroblast proliferation and collagen synthesis. Transforming growth factor beta, TGF-beta, stimulates fibroblast proliferation, collagen synthesis, and angiogenesis. Immunopathology is the study of diseases that result from dysfunction of the immune system. It explores how the immune system can go awry and cause harm to the body instead of protecting it. Key areas of immunopathology: hypersensitivity, exaggerated or inappropriate immune responses that cause tissue damage. There are four main types of hypersensitivity reactions. Type 1, immediate, IgE-mediated, allergic reactions, e.g., hay fever, anaphylaxis. Type 2, cytotoxic, antibody-mediated destruction of cells, e.g., autoimmune hemolytic anemia. Type 3, immune complex, deposition of antigen-antibody complexes in tissues, e.g., systemic lupus erythematosus. Type 4, cell-mediated, T-cell-mediated reactions, e.g., contact dermatitis, organ transplant rejection, immunodeficiency, deficiencies in the immune system, leaving individuals susceptible to infections. These can be congenital, present from birth, or acquired, developed later in life. Autoimmunity: the immune system mistakenly attacks the body's own tissues, leading to conditions like rheumatoid arthritis, multiple sclerosis, and type 1 diabetes. Immunodeficiency. The immune system is weakened or compromised, leading to increased susceptibility to infections. This can be caused by various factors, including genetic defects, malnutrition, certain medications, and infections like HIV, AIDS. Thanks for watching. Hope we provided useful information. Do not forget to share, like, subscribe GNP Sir Classes official YouTube channel. See you again.